Hi, everyone. It's a pleasure to be here today, and I'm going to tell you today about a new approach that we are exploring for achieving optimal metabolic health that goes beyond traditional concept of BMI, but instead incorporates blood measurements into the equation. So first and foremost, I'd like to thank my team. Kengo Watanabe is the first author of the paper and carried a lot of the weight in the analysis that you're going to see today. Tomasz Wilemanski also had tremendous contribution. And of course, our mentors, Nathan Price and Lee Hood. I'd also like to thank the amazing communications, events, and development teams here at ISB that made this seminar possible. So biology is enormously complicated. Systems biology is about understanding how biological molecules interact at the system level. With a recent technological advancement, we now have new abilities to measure what is called multi-omic data. And what it basically means is that we can simultaneously measure comprehensive sets of things that represent different molecular layers within the cell or within the organisms. So these layers, also called omics, this is probably a word you hear a lot, can include genomics. So this is a study of the entire DNA sequence, proteomics. So this is the study of proteins that are big biomolecules that are important to both structure and function, or the study of metabolomics. So this is the study of small molecules or metabolites that take part in our metabolism. All of these data we can now get from a drop of blood. We can measure it longitudinally, so over time, and we can add to it other information, such as data from wearables, from your Fitbits, from electronic health records, etc. And this can give us a wider window into how our physiology, our lifestyle, and our environmental exposures all interact. So I know it sounds futuristic, but with the rates and the costs, at the rate that the cost of measuring all this data drop, the day in which all this data will make their way into the clinic, affecting both preventative and acute care, is very close. So the work I'm going to tell you about today is a study that harnessed multi-omic data to study the heterogeneity or the variability of obesity and its physiological implications. So a little bit about obesity. Obesity is a growing public health concern. According to the National Health and Nutrition Examination Survey, NHANES, over 40% of adults over the age of 20 meet the criteria for obesity. Obesity is a major risk factor for a number of chronic diseases, including diabetes, stroke, cardiovascular diseases, and cancer, some of which are among the leading preventable reasons for premature death. And the obesity is also an economic burden. The estimated annual medical cost of obesity in the US alone was nearly $173 billion in 2019. And the medical costs of adults who had obesity were about more than $1,800 higher than the medical costs for people with healthy weight. And despite despite the common thought, obesity is not only a problem of high-income countries, because obesity is on the rise also at low and middle-income countries, especially in urban settings, which is what you see in this slide. So the most common measure of obesity is probably, you all know, is BMI, or body mass index. And BMI is calculated by a person's weight in kilograms divided by a square of their height in meters. And according to the World Health Organization classification, BMI of between 18.5 and 25 is considered normal, and BMI between 25 and 30 is overweight, and BMI above 30 is considered obese with progressive degree of severity. What some of you may not know is that the concept of BMI was originally developed in the early mid-19th century by a Belgian mathematician and sociologist named Quetelet, who was working on developing a method to describe the average man in terms of correct body proportions. So BMI was not initially created as a tool for assessing health or obesity. Over time, researchers and healthcare professionals observed a very strong correlation that BMI has with chronic disease risk, and began to adopt the Quetelet syndrome, the Quetelet index, as a convenient way to evaluate obesity until the 20th century, when the term body mass index was introduced. And since then, BMI is used as a standard method to screen for potential health-related, potential weight-related health issues. But beyond that, it's used to determine eligibility for certain surgeries and treatments, for screening for clinical trials, and recently, as we saw with COVID, to determine eligibility for a vaccine, for example. But there is a problem. Although BMI is a useful, cheap, and intuitive measure, it is also a crude measure. We have to remember that BMI does not directly measure body fat, and it does not differentiate between fat and muscle mass or between fat and bone density. It also doesn't account for fat distribution. And all of these bone density, fat distribution, and muscle mass have an implication for human health. So, for example, BMI can lead to misclassification of people with high muscle to fat ratio, for example, athletes. So, in fact, studies show that BMI misclassifies individuals in about 30% of the cases. And that's a huge problem. It means that almost 1 in 3 people that have a normal BMI are in fact at an elevated risk for chronic diseases. And what is more worrisome is that they are most likely not perceived that way by their clinician. So because obesity is such a heterogeneous and complex condition, in order to improve our understanding of obesity and its impact on health, we decided to use blood as a window, like we like to say at ISB, and see if we can calculate BMI not based on height and weight, but based on comprehensive blood measurements, because we wanted to cast a wider net and to identify a broader range of potential biomarkers, and with the hope that some of which may be amendable to interventions. So, just to mention that our study builds off previous research, and the most relevant one is from 2019, and that's what you see in the slide. Cerulli and colleagues used blood metabolomics, so the small molecules that we can quantify from the blood, and they combined it with whole genome data, whole genome sequencing data, to identify metabolic and genetic signatures of obesity. And they found that the metabolome could capture different aspects of obesity. They were able to explain 49% of the variation in obesity by blood metabolome. And I want to take a second to explain what it means. It means that 49% of the changes in BMI can be attributed to changes in blood metabolites. And the rest of the percentage can be explained by lifestyle, diet, or anything else. So remember this number, sorry, 39 actually, I misspoke. So remember this number 39, and the performance was much better than any combination of standard clinical markers that they try to use. that like LDL cholesterol, total cholesterol, triglycerides, and so forth. And it was better than any single biomarker. And so it seems that although single biomarkers that we like to use, or single metrics like BMI, can be very useful, if we create a signature from multiple types of measurements or multi-omics data, it has the potential to bridge the gap between what BMI can capture and the heterogeneity of obesity. So with new technologies that have evolved, with richer data sets and with different analytical methods, we set out to study obesity using a large cohort of multi-omic data. So in order to do that, we leverage data from participants in the Aireville cohort, which is a description of which you can see on this slide. Aireville was a consumer-facing wellness program that operated between 2015 and 2019, and they collected the wealth of data on their participants. Most of the participants in the program agreed to that their de-identified data will be shared and analyzed for research purposes. So for this study that I'm talking about today, we selected participants from the Aireville datasets who had BMI measurements and all the omics data that we wanted to analyze. So metabolomics, proteomics, clinical labs, and genetics. The size of the cohort was around 1,200 participants, and you can see at the upper, you can see at the upper, at the bottom plot, the spread of the ages, and in the upper plot, the spread of BMI. But essentially, we had more females than males. The average age was just below 50, and most of the participants were white with mean BMI of around 28, which is overweight. Overall, the people in our cohort were not screened for any particular condition. So they were generally healthy in a way that was representative of the populations that they were drawn from, that were, this was mainly from Washington and California. So overall, the number of blood measurements that we were looking at was more than 1,100. And one more thing to note here is that in the Aireville program, participants received healthy lifestyle coaching with the goal of optimizing their health and wellness. And the next sets of analysis that I'm going to show you are based on their baseline measurements, but keep in mind that there was an intervention, because we will get to that when I describe the longitudinal analysis. Okay, so as I mentioned, we wanted to obtain a system-level understanding of obesity and try to calculate BMI from internal measurements that are potentially closer to the physiology. So in order to do that, we used machine learning to create what we call biological BMI scores. Those are BMI values that were calculated from blood measurements. And we calculated several such scores. So one from a set of standard clinical labs and demographic data, such as that are listed here in the top, like sex, age, cholesterol, glucose, insulin, and so forth. And that's what you see on the top gray panel. And then 4 measures using the multi-omics data that we had. So one based on metabolomics data, that's the blue one. One based on proteomics data, that's the red. One based on a very wide panel of clinical labs, that's the green one. And one that combines all of them, that's the purple one. So for every participant, we had their classical BMI, which is what you see on the vertical axis of these graphs. And we also had their predicted BMI that was based on the models, which is what you see in the horizontal axis of these graphs. So for example, the left plot shows how biological BMI is computed from blood metabolomics. And it shows the correspondent between metabolomics BMI and classical BMI. And all of our 4 omics models at the bottom did much better than the gray model at the top. That was based on the standard clinical labs. And you see that by the fact that what I listed and the percentage that I listed is the amount of the variance or the changes in BMI that our models were able to explain. So overall, across all the 4 models, the models retain 132 parameters, and they were able to explain up to 78% of the variance in BMI, while the clinical labs, and that's interesting because this is what you get when you go to the doctor. So that had the worst performance, 48%, and that was the widest clinical labs that you don't normally get but exists in service providers. But the best ones were the ones based on all the omics and the ones based on metabolomics and proteomics alone. So to confirm the generalizability of our results, we validated our analysis in data from the Twins UK registry, which is an external data set. And what we confirmed is that when we train a model on the omics data, again, also in the Twins UK data set, it did much better than a model that was trained on standard clinical labs. So this suggests that a large portion of the metabolic perturbation that are caused by obesity is captured by the plasma metabolomics and proteomics beyond the widely used clinical labs. So next, we wanted to verify that the biological BMI maintains similar associations to the classical BMI. For example, with body shape, with genetics, for example, high genetic risk for LDL cholesterol or BMI, with lifestyle, for example, sleep and activity, and with physiology, for example, blood pressure. So looking at 51 features that we had in the data, in 27 of them, which is what you see in this slide, we discovered significant association and with some small differences in all the omics-based biological BMI had the same correlations with these features as the classical BMI. So for example, high BMI was associated with high waist to height ratio. That's the green arrow. It was also associated with high blood pressure. So that's the orange arrow. It was associated with high LDL cholesterol. That's the black arrow. And it was also associated with sedentary lifestyle, and that's the purple arrow. So these results support the hypothesis that the biological BMI model basically maintains the same characteristics as the classical BMI. But next, we decided to look at the people for which the obesity category based on classical BMI deviated from the obesity category based on their biological BMI. So for example, people who were classified into normal BMI category by classical BMI, but into obese BMI category by biological BMI, and vice versa. When this happens, we call it misclassification. So the y-axis, the vertical axis in this plot, shows the misclassification rate, and we calculated this rate for each of the types of biological BMI. And those are the different colors. And we did this separately for every weight category of classical BMI. So to make a long story short, what you see here is that approximately 1/3 of the participant, if to be more accurate, between 30 and 50% of the participant were misclassified by biological BMI, meaning that they had a different obesity category between the classical BMI and the omics BMI. And that matches what is known from the literature about the rate of misclassification by classical BMI. But what does this misclassification mean? Does it mean that BMI identifies those 1 in 3 people that classical BMI misses? Do people with normal BMI but obese metabolic BMI have worse health than people with normal classical BMI and biological BMI? So in order to test that, we look deeper into these unclassified or misclassified groups of people whose biological BMI class didn't match their classical BMI category. The trend with the misclassified people was that people with normal classical BMI, if they were misclassified by biological BMI, it was usually into the obese category. On the other hand, the people with the obese classical BMI, if they were misclassified by biological BMI, so they tended to be predicted to have a normal biological BMI. So it looks like biological BMI is able to capture both the people with healthy weight, but more the people with healthy obesity, but more importantly, it's able to capture the ones that have normal BMI, but supposedly unhealthy metabolic health. So in order to know if this is indeed true, we wanted to look at the difference between the biological BMI and the classical BMI for healthy and unhealthy people in the different BMI categories. So since there is no one accepted definition of healthy, we decided to look at it in several ways. And what you see in this slide is where we categorize people to healthy and unhealthy, which is red or gray, based on satisfying at least 2 of the criteria for having metabolic syndrome, according to the National Cholesterol Education Program. And I listed these 4 criteria in red. So now let's look at the left plot, for example, with the title Metabolic BMI. In the left side of this figure, we see the data for people with normal classical BMI. So we see that out of these people with normal weight, the ones that are healthy, the ones that are healthy also had a normal biological BMI. So the difference between the biological BMI and classical BMI, and that's what you see in the vertical axis, is closer to 0. However, the unhealthy people with normal weight, with normal classical BMI, had a higher biological BMI. On the other hand, when we looked at people with obese biological BMI, and that's the blue rectangle now, the healthy ones also had a high BMI, but the unhealthy ones also had a high biological BMI. But the healthy people in the obese category, so the healthy obese, indeed had a lower metabolic BMI. When we looked at the activity parameters or lab results of these individuals, the results made sense. So for example, people with normal classical BMI that had an obese biological BMI also had significantly higher waist-to-height ratio, resting heart rate, higher blood pressure, lower daily activity, and so forth. When you compare it to a matched group in the obese category. And then this all suggests that biological BMI is able to capture these cases where classical BMI misclassifies people. So biological BMI is able to identify those people with normal weight, but poor metabolic health that may be unaware of their risk for disease. So for the next point, I'd like to take a little segue into one of our previous projects that was published several years ago in the Journal of Nature Biotechnology. And this was a joint work in collaboration with Tom Wilmansky in the lab and several other collaborators, including the Gibbons lab at ISB. So just to give you a little bit of background, the gut microbiome is linked to a number of diseases from IBD to cancer. And there are several ways in which the gut microbiome can affect our physiology through its influence, for example, on nutrient absorption, through metabolism of diet components, and by modulating development of the immune system, etc. However, determining what a healthy microbiome is not trivial. And that's mainly because the abundance and the presence of specific microbes has been shown to change across different geographic locations, different environments, and different diet types. Despite that, higher gut microbiome diversity is considered healthy, and low microbiome diversity is considered is associated with several diseases. So the goal of this project was to look into the extent to which the gut microbiome diversity is reflected at the molecular level in the host itself through the blood measurements. And just to summarize the finding in a short way, so we were able to show that 45% of the variation in gut microbiome diversity can be explained with blood metabolomics, but cannot be explained by a wide panel of clinical labs or by the proteomics data, a set of proteins that we could measure. We discovered a signature of 40 plasma metabolites, 13 of which of microbial origin, that composed the signature that was able to predict gut microbiome diversity. And an important suggestion from this work is that the blood metabolome could be a more robust and universal predictor of gut microbiome health than actually quantifying the specific species in the feces. So I wanted to show this project because, it's interesting, but also because it's another way where we can use blood multiomics data for monitoring and for prevention in the context of gut health. Back to our original biological BMI story. In the paper that I just told you about, we found that BMI explained an order of magnitude less variance in gut microbiome diversity than a set of the set of 40 metabolites that we discovered. And therefore, we hypothesized that our biological BMI, and specifically our metabolomics BMI, would have a stronger power or stronger association with gut microbiome health and diversity than classical BMI. And as we hypothesized, we observed that biological BMI is a stronger predictor of gut microbiome diversity than classical BMI. So what you see in this graph is that overall, gut microbiome diversity is decreasing with increasing BMI. But more specifically, the metabolomics BMI is the strongest predictor of gut microbiome diversity. We see that people with normal classical BMI that had an obese metabolic BMI have lower gut microbiome diversity than people that have matching classical and biological BMI, and vice versa. People with an obese classical BMI but normal biological BMI also have higher gut microbiome diversity. So the take-home message here is that metabolomics BMI more strongly reflects gut microbiome health than observed BMI. So next, we attempted to use the microbiome composition, the levels of the different bacteria, to predict the obesity category of participants using machine learning algorithm called Random Forest. So what you see here is a plot that we use a lot to evaluate the performance of machine learning model. It's called a ROC curve. And the take-home message here is that gut microbiome composition was able to better predict the obesity category based on biological BMI, and this is the blue line, then it was able to predict the obesity category based on the classical BMI, and that's the solid black line in the plot. So this reinforces our previous results that biological BMI, and specifically the metabolic BMI, properly reflects the microbiome health in terms of diversity more than classical BMI. And as in the previous results, we validated this external this result in the external Twins UK cohort, which is what you see on the right. So far, all the data that we have looked at was baseline measurements. But remember that people in our cohort underwent lifestyle coaching to optimize different areas of their health. So next, we decided to perform longitudinal analysis, meaning like a time course analysis, to see how changes in biological BMI correspond to changes in actual BMI. So we analyzed 608 participants that had longitudinal data in our cohort. And what you're looking at here is the estimated average change in classical BMI in black, metabolomics BMI in blue, proteomics BMI in red, and clinical labs BMI in green. And what we see here is that classical BMI, again, that's the black line, was decreased in 6 months, 6 months into the program, and then increased back up 12 months after the enrollment into the program, which is consistent with the previous paper that is shown in the bottom, that also shows this pattern of weight loss until 6 months and then regain at 12 months. Now, biological BMI is also decreased during the program, and the strongest effect is shown in the metabolomics BMI. So that's, again, the blue line. So we see that metabolic BMI decreases sooner and stronger than classical BMI overall, and this was even more extreme if we do the same analysis only for the people with baseline normal classical BMI, which you see on the right. So another interesting thing to note here is how the different biological BMI types respond where biological BMI that is based on clinical labs or proteomics data improved much slower than both metabolic BMI and even the classical BMI. And this different dynamics in response is interesting because it can say that we're just not measuring the relevant things, or it can indicate that those different modalities change at different rates. So clinical labs just respond slower to changes in your metabolic health. So it is well known that people with higher BMI have an easier time losing weight. And it was previously shown by our own team several years ago that individuals with obesity had a larger drop in BMI while on the Airvel program. So the final set of interesting results that I like to present today is that metabolic BMI is also more informative about weight loss potential and metabolic health improvement than actual BMI. And again, we used a method called linear mixed modeling to calculate the average change in BMI while accounting for covariates, as I forgot to mention, was also done in a previous analysis, sex, age, season, etc. And in this graph, the horizontal axis shows the time in the Airvel program during which the people received the lifestyle intervention. And the vertical axis shows the average percent change in BMI. So the left plot, which is what you see now, shows that the people that had normal classical BMI at baseline. Then we divided the people with normal BMI into two groups. Those that had the same BMI category according to both the classical and metabolic BMI, and that's the blue line, and those that had a different BMI category based on the metabolic BMI in orange, which I showed before, was usually higher. So these are the people with normal BMI, but obese metabolic BMI. In this case, the people that were misclassified by the biological BMI to be obese, again, the orange line, were able to decrease their metabolic BMI at a higher rate than people that had normal BMI and weren't misclassified. So they also had normal metabolic BMI. And that despite the fact that many of these people saw only no or only a minimal change in their weight. On the right side, you see a similar result for the people who had an obese baseline classical BMI. So those that had normal metabolic BMI, which are sometimes called the people called healthy obese, this time they were able to improve their metabolic BMI less, and that's the orange line. And the people that had obese classical and obese biological BMI were able to improve their metabolic BMI more while on the program. So overall, this data suggests that metabolic BMI estimates better reflect not only the metabolic health, but also weight loss potential and metabolic health improvement potential better than actual BMI. So to summarize, I showed you today that I told you today about a little more than what you probably knew about traditional BMI, that it's a simple metric that is derived from height and weight. It's so powerful because it correlates with a number of chronic diseases, and although it's undoubtedly useful, it fails to capture the heterogeneity of obesity, and it misclassifies people in about 30% of the cases. I also presented our newly developed sets of biological BMIs that were calculated from the blood multiomics data. And those measures integrate molecular data that we can capture from drop of blood. They better reflect metabolic health and are more tightly associated with gut microbiome health. And they're able to identify people that have normal BMI, but the 30% of people that have abnormal BMI and normal BMI, but abnormal metabolic health. And they're also more responsive to lifestyle changes. So what are the implications of this study? So our study substantiated how obesity basically affects every aspect of our physiology. We showed that biological BMI captured this variability, and it can identify misclassified individuals. And I want to stress again, this is specifically important for those with normal BMI but abnormal metabolic health. And those people are probably right now may not be properly treated based on their risk. And because biological BMI, specifically the one calculated based on metabolomics data, is more responsive to lifestyle changes, it offers a way to give people valuable feedback and to monitor the effects of health interventions and lifestyle changes. And this can be crucial in keeping people engaged in lifestyle changes, because we all know that, sometimes we make a lifestyle change and we don't see our weight budge, and then we kind of stop. But if we could, keep seeing that we are continuing to benefit from the lifestyle change, that would motivate us to continue. And although weight loss success rates are very low following lifestyle changes, there is evidence that even intermittent weight loss can have an effect on insulin resistance and chronic disease, even 20 years into the future, even if the weight was regained. And the fact that we saw that our metabolic BMI remained low even after the weight was regained, although we had a very short follow-up of a year, we are hopeful that in future studies, maybe we could show that metabolic BMI also captures this health benefit in intermittent weight loss, also for a longer time span. And in the broader sense, our study highlighted the potential of multi-omic data in precision medicine. And as more and more value is shown for deep molecular profiling, such as this study, we strongly believe that this will become the standard of the trade in medicine, and will accelerate the use of this kind of scientific wellness-based, data-based metrics that in precision medicine, also in the clinic. That's it.
Thank you, Noah. Can you hear me?
I can hear it.
Great. So we've already got a bunch of questions. So we'll try to move through as many as we have time for. The first one, we'll start simple. How do you plan to continue this research?
Yeah, so we have a number of directions where we plan to continue the research. We plan, as I mentioned, to assess the predictive power of biological BMI and its association with long-term outcomes. So is biological BMI more predictive of chronic disease and all-cause mortality than classical BMI? When we look at longer time span than the follow-up we had in the Airval data set. We also intend to look more closely how genetics play into it. What are the differences between males and females? Our cohort was based on primarily on white individuals, so we plan to try to get more data from more diverse cohorts. And we also plan to compare the biological BMI to a concept that was also developed by our lab and other labs of biological age. So what is the difference between predictors of biological age and biological BMI?
Great, thank you. So next one, how are people with normal weight that are metabolically unhealthy different than healthy normal weight individuals? From your talk, it seems it can't be all seen in standard annual clinical labs.
Yeah. So studies found that people with metabolically unhealthy normal weight have a different fat distribution than people with metabolically unhealthy normal weight, and also the metabolically healthy obese. So these people with the metabolically unhealthy normal weight have a low amount of gluteofemoral fat mass, which is fat that is stored beneath the skin, predominantly in the hip and thigh region. So data from other studies show that low gluteofemoral fat mass is associated with higher risk of chronic diseases in lean women. That was study done in the Women's Health Initiative dataset. So it seems that metabolically unhealthy normal weight individual store less of their fat around the hip region, which is considered a healthy sink to store excess fat.
Interesting, thank you. So another one, how can I determine my biological BMI? Is this something my doctor can do? If not, when will biological BMI be available to the public?
So currently, this is a research project. This is still at the level of the basic research. We are hoping that as these measurements become cheaper, we are able to team up with, either a private company that is going to sell this product, or hopefully with, with clinical care providers or insurance company that would be able to fund this for you. But currently, there's no way for you to get this data because it's not commercial, to get your biological BMI because it's not commercialized yet.
Okay. In the future. Another question, very interesting results. Other than BMI, how about waist circumference? Are there specific reasons not to go for that? Secondly, are effect alleles added into biological BMI as variables? And you might want to tell the lay folks what effect alleles are.
Effect allele. I'm not sure I know myself. So if the person
Okay. Well,
effect of what? If you mean the ancestry, then yes. If you mean the effect of, the polygenic risk score effect sizes, then we determined that there was no difference in the polygenic risk scores between the different categories of misclassified individuals. But I'm not sure what this question means. But for the other part of the question, in general, fat distribution in the body is an important feature for understanding obesity. And in particularly to your question, obesity around the central area of the body or abdominal obesity is associated with chronic diseases and like metabolic syndrome. And I'm imagining this is why you're asking this question. So we didn't have hip circumference data. So instead of waist to hip ratio, so this is what people usually use to normalize for body size. So we calculated waist to height ratio. And indeed, we've built like biological waist circumference models. And but this model showed a lot of overlap with the biological BMI models in terms of both their associated measurements, their the health status of misclassified individuals, and all the analysis that I've shown. And moreover, in our validation cohort, the Twins UK cohort, they had DEXA scans. So those are able to quantify fat distribution. And we found that fat in the android region, and that's around the waist, was significantly higher in people that had normal classical BMI, but abnormal metabolic BMI or all the types of biological BMI. So it seems that omics-based BMI, that the ones that we have calculated, are accounting for fat distribution as well as abdominal obesity. So that's why we chose to concentrate our paper on BMI rather than on waist circumference measure.
Got it. And you kind of already answered this one, but I'm going to read it for the compliment. So glad to see this research, as I have always contended that muscle mass, fat distribution, and bone density needed to be considered, but this is well beyond that. And then how long for this to be integrated into our healthcare system, which I think you already kind of
that's a tough question. It's expensive right now, but the costs are dropping. And we are hoping that, in 1, 2, 3 years, the costs will be low enough and we will have shown the benefit. And, one of the analysis we still need to be doing is to show how this is saving healthcare costs. And that might be useful in convincing, kind of insurance companies to fund this. And this will, it's a cycle, this will lower the cost even more and so on and so forth. So this is the future, no, undoubtedly, but the future is not here yet.
Okay. What are some of the proteins or metabolites that were important for calculating the biological BMI? Were there some surprising ones?
So what's interesting about our study that we identified a total of approximately 120 both metabolized proteins and clinical labs that were important to calculate biological BMI, across our different models. Not every model uses this number, but some of these were analyzed that were known to be connected to obesity and disease risk, and some were either not known to be related, or in case of many metabolites, those were just new compounds that were not measured before. So some of these known analytes that had positive association with BMI included leptin, so it's the hunger hormone, adrenomedulin, fatty acid binding protein 4, as well as elements in the immune system that have pro-inflammatory effect, such as interleukin 6. And homoarginine, and that's a metabolite known to be a biomarker for cardiovascular disease, was also a positive predictor of biological BMI. So that's interesting because we identify both things that are associated with obesity, but also things that are associated with disease risk. And other thing that were discovered, for example, are different forms of lipids, such as sphingomyelins. And those are found in cell membranes, in structures called lipid rafts, and they are important for signal transductions between outside and inside the cells, and uric acid. And interestingly, some of the species of lipids were negatively associated with high BMI. And that's something that is an interesting finding. Another interesting predictor was a protein called RAGE, which was previously associated with type 2 diabetes and cardiovascular disease. And that was also, again, it was associated with the risk, and it was also a strong predictor of, or a strong component of biological BMIs.
So your study mainly analyzed white people. Is there a reason to believe that the model will be different in other ethnicities?
Yeah, the reason our study analyzed white people is that this is what we had in our cohort. Both the Twins UK cohort and the Arable cohort are very predominantly white, and we could not get multi-omics data from more diverse cohort at this point, but we are pursuing that. And there is a difference in the prevalence of obesity in different races. The highest is in Blacks, and then followed by Hispanics, and then non-Hispanic white adults, and finally Asians have the lowest obesity rate. But since the metabolic BMI, for example, relies on metabolites in the blood, many of which are, as I explained, metabolites by the gut microbiome from our food. So it is reasonable to think that in different ethnicities that potentially have different exposures and different diets, and that it will be reflected differently in the blood metabolites. So the biological BMI scores would have to be different.
And you mentioned that there might be benefits even if there's weight loss and the weight's regained, that the benefits may last. Is there evidence that you can benefit from even a moderate weight loss? Can you talk about that?
So this is not something that we investigated in this study, but studies showed that moderate weight loss of even 5% improved the multi-organ insulin sensitivity. So the ability of our cell to respond to the insulin hormone and reduce blood glucose levels, and also in beta cell function, the ones that secrete the insulin. And if additional weight loss also improved beta cell function, insulin sensitivity in the muscle, and that kind of, so yeah, there is a lot of benefit to even weight loss as low as 5%.
Good to know. Okay, I think this needs to be our last question. You said that biological BMI explains X% of the variation in BMI. What does the percent explained variation mean?
Okay, so this, as I said in the talk, it basically means that how much in the change in one variable can be attributed to change in another. So for example, if a study finds that exercise explained 40% of the variation in weight loss, it means that 40% of the differences in weight loss among individuals can be attributed to their exercise habits. But the remaining 60% of the variation can be due to other factors like diet, genetics, or other lifestyle choices. So it's important to note that this relationship does not necessarily imply causation, but it does show that there is a correlation between the two variables. So in other words, while the relationships suggest that the two variables are connected, it doesn't necessarily mean that one actually causes the other.
Got it. Well, thank you so much for a terrific presentation and for taking all those questions. It was great, Noah. And thank you all who joined us today.